Hello everyone, I am Dr. Sharad Deshmukh. You are watching my channel Microbial content. We are studying sterilization and disinfection. In sterilization and disinfection, we have already started the various methods of sterilization. So under that we have seen the physical agent, and today we are going to start the chemical agent. And the method in which we are generally using the chemical agent, the process is referred as the disinfection. We have already defined the term disinfection. So in which the chemical agents are used which kill the pathogenic bacteria, that is the organism which are killing the bacteria causing the diseases. That process we have defined it as a disinfection. For the process of disinfection, there should be the ideal disinfectant, and for the ideal disinfectant they should have the ideal various properties they should possess. One of the most important is the first one is the wide spectrum of the activity. So here the term itself suggests they must have the wide spectrum, so they should act on the both the type of the organism, that is gram positive as well as the gram negative, and the other microorganisms also. Secondly, it should be active in presence of organic matter. This is also one of the most important character when we are using this chemical agent. So if you are adding on the certain surfaces or on the particular area, even though the organic matter is present, they should able to act on the microorganism. The next character, it should be the stable. It should remain stable for the longer period. Then it can act for the longer time. So that is also one of the most important property they should possess. They should be the stable one. The next one is it should be effective in acidic as well as the alkaline condition. If they are acting in presence of any organic matter, there may be the change in the pH on the surface, but our agent should be able to act in acidic condition as well as the alkaline condition. Then and only then it will able to kill the most of the microorganism. Next character, they should have the speedy action. So when the agent is applied on the surface or any object, they should act very rapidly so that they are able to kill the microorganism very rapidly with the high speed. So they should have the speedy action. The next character they should possess, they should have the higher penetrating power. So if you are applying the agent on the surface and they are having some already component on the surface, they should able to penetrate, and they should able to penetrate up to the microorganism and should be able to act on them. So they should have the high penetrating power. Then and only then they will act on the microorganism. In addition to this, they should also have the other properties, like they should be non-corrosive, otherwise may cause the harm. They should not be toxic one, otherwise if it is a toxic one, it is difficult to use this agent. Then they should not cause the local irritation. If they are able to cause irritation, it will be the harmful one. They should possess this most important property. Then they should be inexpensive and easily available. This is also one of the most important property, particularly for the developing countries. It should be less expensive, and most importantly they should be easily available so that that can be used regularly. Then they should be safe and easy to use. This is also not last, but the most important character, they should be the safe and easy to use in the process of the disinfection. Now we will see the next property, that is the factors which are determining the potency of the disinfectant. What are the different factors they can determine the disinfectant is powerful or not? The first one, there are concentration. So it must be used in the appropriate concentration, then and only then they will able to kill the microorganism. So appropriate concentration should be used. So the concentration also determine the potency of the disinfectant. If in very dilute form, it may not be able to kill the microorganism, but if it is used in the appropriate concentration, it will be able to kill most of the microorganism. The second important factor is the time of action. The certain time period is required to kill the number of organisms, and they should be act rapidly in the short time, and that time period is referred as the time of the action required to kill the microorganism. The third character is the pH of the medium, that will also determine the potency. So we have already seen in the earlier part, the pH will also play the role. So your agent must be able to act at alkaline pH as well as the acidic pH, and that pH should not interfere with the action of this agent. Then it will be the more potential. The next is the temperature. The temperature is also one of the most important factors that determine the potency of the disinfectant. Certain disinfectants which are highly vaporizing, they may not be able to act at the high temperature. So the temperature factor determine the potency of the disinfectant. Certain disinfection may require some optimum temperature to act. A very high temperature may not be sufficient for this disinfectant. The next one is the nature of the organism. This is also most important character. The most of the disinfectant are able to kill most of the vegetative form as well as some viruses or some fungi and other microbial agents. But there are some agents which are able to act only the bacteria, or some may able to act the viruses also. And second aspect is, as far as the nature is concerned, the disinfectant are mainly acting on the vegetative cell. They are able to kill the vegetative cell very easily, but all disinfectant are not able to kill the spore of the bacteria. Very few disinfectant can act on the spore. So the nature of the organism also determine the potency of the disinfection. The next one is the presence of the extraneous material. If any additional material is present where you are applying this disinfectant, that may interfere in the action of the disinfectant. But if the disinfectant is the powerful, it is the potent one, it may be able to act even in presence of the extraneous material. It can penetrate rapidly and will act on the cell. Then the most important aspect is the mechanism of action of this disinfectant. These chemical agents, that is disinfectant, can act in the various ways. These are the various mechanisms by which they can act, that is by the protein coagulation, they coagulate protein of the cell. Then they may disrupt the cell membrane, acting on the cell membrane and will able to kill the cell. They are also able to remove the sulfidyl group in the cell and will interfere with the cell metabolism. The next important is they may compete with the substrate, that is substrate competition, and if they are able to compete the substrate, they will able to kill the bacteria the more easily. Now we'll see what are the various chemical agents which are used as a disinfectant. You can see here the list of the various agents which are commonly used as the chemical disinfectant. The first one most important is the alcohol, the aldehydes, dyes, halogens, phenols, surface active agents, metallic salts. These are most commonly used agent. And another group, it is separately mentioned as the gases because they are used in the gas form. So that's why we have mentioned separately at the last. Although these are also the chemical agents. In some book, you may come across the term acid and alkalis. Acid and alkalis are the chemical agent which are able to kill the spore, but they are difficult to handle. That's why nowadays it is not mentioned in the most of the books. So it is mentioned the list, that is alcohols, aldehydes, dyes, halogens, phenols, surface active agents, metallic salt, and the gases which are most commonly chemical agents used as a disinfectant. The first we'll see about the alcohol, and among the alcohol, the most commonly used alcohol are ethanol and isopropyl alcohol, which are commonly used. And one third agent in the alcohol is the methyl alcohol. But first we'll see about the ethanol and isopropyl alcohol. These alcohols, they don't have the action on the spores. We have already mentioned that these agents are not necessarily to kill the spore. Some may be able to kill the spore, but alcohols are not able to act on the spore. And second factor, we are also seeing the concentration. So we should use the optimum concentration of the agent, and the alcohol is able to act in the concentration between 60 to 90% in the aqueous solution. But generally the 70% of alcohol is commonly used in the aqueous solution, and that is able to act on the microorganism, that is on the bacteria, and able to kill them. And the mechanism of action of alcohol is mainly by denaturation of the proteins in the bacteria. So they act by denaturation of the bacterial protein. If it is used in the very high concentration, they may not able to act, because that will result into the dehydration of the cell, and it will not able to penetrate through the cell. That's why the optimum concentration must be used. Here it is mentioned the range 60 to 90% in aqueous solution, and I have already told that the optimum concentration commonly used is the 70% of the alcohol. So in this case, the first we'll see about the ethanol, which is most commonly used agent among the alcohols, and it is commonly used as a disinfection of the skin. So during the vein puncture or the any injection and by the physician or the surgeon, they apply the ethanol on the surface of the skin. That is for disinfection of the skin, and that is the commonly used as a ethanol in the 70% concentration. The second one is the isopropyl alcohol. This isopropyl alcohol is the commonly used as a disinfection of the clinical thermometer. We will observe that the clinical thermometer is kept in the bottle containing the isopropyl alcohol. So as it will act as a disinfectant, and that will disinfect the clinical thermometer which is used by the physician or the surgeon. Then in the alcohol, the isopropyl alcohol is preferred as compared to the ethyl alcohol. Why? Because it is better fat solvent, it is more bactericidal and less volatile, and these are the properties which are the better than the alcohol, that is the ethyl alcohol. Main important we have mentioned first, that is the better fat solvent. It will rapidly solubilize the fat in the bacterial cell, and that's why they are having the more bactericidal action as compared to the ethyl alcohol. And the most important is the less volatile. That's why it will remain for the longer period. Otherwise, if it is highly volatile, it will not act for the longer period. So this property is useful for the isopropyl alcohol. The last one, that is the ethyl alcohol, is mainly effective against the fungal spore, and it is mainly used for treating the cabinets and the incubators. It is applied to the cabinets and incubator to destroy the fungal spore. They may generally interfere, particularly in the incubator. They cause the contamination of the media, or in the inoculating chamber they cause the contamination. So it is generally used for treating these cabinets and incubator to destroy the fungi which interfere in the isolation of the organism. But one of the most important aspect in methyl alcohol is the methyl alcohol vapors are highly toxic and it is inflammable. So it must be handled very carefully, and that is one of the disadvantages you can say for the use of methyl alcohol, as methyl the vapors are the very toxic and it is inflammable. Now we will come to see the second most important agent, that is the aldehydes. If aldehyde is concerned, the most two commonly used aldehydes are the formaldehyde and the glutaraldehyde, which are frequently used as a disinfectant. And these are mainly acting against the amino group in the protein molecule of the cell. We have already seen that the earlier one, that is the alcohol, was able to coagulate the proteins of the cell. Now here the aldehyde is active against the amino group in the protein molecule, and this is the mechanism by which it acts. The formaldehyde is the bactericidal, sporicidal, and has a lethal effect on the spore. This is one of the best character of the formaldehyde among the aldehydes, that is, which is active against the bacteria, against the bacterial spore, as well as it is also having the lethal effect on the viruses. And that's why it is one of the most important disinfectant used in the microbiology, that is the formaldehyde. Now we'll see the various uses. So it is used to preserve the anatomical specimen. Everybody know that in the laboratory, the various specimens are preserved in the formaldehyde, particularly the 10% concentration of the formaldehyde, so that they remain free from the microorganism. Otherwise, if they are kept as it is, they get destroyed by the putrefying action of the microbial agent. So they are preserved in the formaldehyde. The second one is to destroy the anthrax spore in hair and wool. This formaldehyde is able to kill the anthrax spores, and that's why it is commonly used for the disinfection of the hair and wool. And because the particular anthrax bacilli are present or the causing the infection from the sheep, and the wool is prepared from the hair of the sheep, and they must be subjected to the action of the formaldehyde to kill the anthrax spore. The next one is the combination of the 10% formalin and 0.5% tetraborate is used to sterilize the clean metal instrument. For sterilization of the cleaned metal instruments are mainly by using formalin and a combination with the tetraborate, which is commonly used. Next is the formaldehyde gas in the gas form is used for sterilizing the instruments and the heat sensitive catheters. Mainly here you can see here, as this chemical agent may cause the certain harm to the particular instrument or certain material what we are sterilizing or disinfecting, but the formaldehyde in the gas form is commonly used for the instruments, and particularly the heat sensitive catheters, they are subjected to the treatment with the formaldehyde in the gas form. The next important agent in the aldehyde is the glutaraldehyde. Glutaraldehyde is effective against the tubercle bacilli, fungi, and viruses. Here also it is also the powerful chemical agent, where it is active not only against the routine bacteria but also against particularly the tubercle bacilli, as well as the fungi, as well as the viruses. So it is having the broad activity, and that is definitely used for the destruction of these all microorganisms, and it is less toxic and irritant to eyes as compared to the formaldehyde. This is also the one of the advantage over the formaldehyde, that is the less toxicity, it is less irritant to eyes as well as to the skin as compared to the formaldehyde, and it is used to treat the corrugated rubber anesthetic tubes, face mask, plastic endotracheal tubes, metal instrument, and polythene tubings. Particularly, it is useful mainly in the hospital, because these the agent we are the what we are mentioned here, the uses for rubber anesthetic tubes, face masks, and these are all are used in the hospital. And one of the most important aspect is they are more effective against the tubercle bacilli, fungi, as well as the viruses. So they are having the broad activity, and that's why they are the better killing agent against this number of microorganisms as compared to the other chemical agent. The next one we will see about the dyes. As far as the dyes is concerned, we come to know about only the staining method, where we are using mainly the two types of the dyes. There are two groups. The first one is the aniline dyes, and second one is the acridine dyes. As far as the dyes is concerned, we come to know about the staining of the bacterial cell. But in addition to this, they are also having the activity against the bacteria. First, we will see about the aniline dyes. As far as the aniline dyes are concerned, we are commonly using in the laboratories are the brilliant green, malachite green, crystal violet, basic fuchsin. Everybody is well known about this staining solution, that is the dyes, and we are commonly using in the laboratory. But we will only think about for the staining purpose. But as these dyes are also active against this bacteria, particularly they are highly active against the gram positive bacteria than the gram negative. And that's why these dyes are also used in the media. You can see here there are no activity against the tubercle bacilli, so the malachite green is used in the LJ media. Now, if the malachite green is added into the Lawson-Jensen media, which is used for the cultivation of Mycobacterium tuberculosis, and there are no activity against the tubercle bacilli, and so that they will able to inhibit the other bacteria and they will promote the growth of the tubercle bacilli, and the other bacteria will not interfere in the growth of the tubercle bacilli in the LJ medium. That's why these dyes can be used as an selective agent. Then secondly, it also helps as a selective agent in a solution isolation of the gram negative bacteria. Here we have already mentioned they are very active against the gram positive bacteria, and these aniline dyes can be incorporated in the media for the isolation of the gram negative bacteria, because they will inhibit mostly the gram positive organism and help to isolate the gram negative bacteria. They are also having the lethal effects on the bacteria, and that is due to mainly the reaction with the acid groups in the cell wall. This is the mechanism by which the alanine dyes act, that is they are in the lethal effect, and they are having the reaction with the acid group in the cell wall. They are interfering with the cell wall. The next one we'll see about the acridine dyes, and these dyes are mainly orange in color, and these acridine dyes are mainly effective or highly effective against the gram positive than the gram negative. So definitely it will easily able to kill the gram positive bacteria as compared to the gram negative. That effectiveness is more against the gram positive bacteria. And the important dyes which are included in the acridine group are the proflavin, acriflavin, euflavin, and aminacrin. These are the various dyes which are under the acridine group. And the lethal effect of these dyes is mainly due to the reaction with the acid group of the cell. They are having the reaction with the acid group, and again they may impair the DNA complexes of the organism, and thus they are able to kill or destroy the reproductive capacity of the cell, mainly with interfering with the DNA complexes of the organism. And now we'll stop here for the disinfection process in as a first part, and thanks for watching my video, and more content will be posted on this channel in the coming lecture, the remaining part of the disinfectant. Please like, share and subscribe to my channel. Thank you very much.